Life requires energy. This energy is used for various cellular processes. These processes include muscle contraction, nerve impulse transmission, and molecule transport. Adenosine triphosphate, or ATP, is the primary energy currency of cells. It stores energy in its chemical bonds. When these bonds are broken, energy is released. This energy can then be used to power cellular work. Think of ATP as a rechargeable battery. When the battery is fully charged, it has a lot of energy. This energy can be used to power devices. Once the energy is used, the battery becomes discharged. Similarly, ATP can be hydrolyzed to release energy. This process forms adenosine diphosphate, or ADP, and inorganic phosphate, or Pi. Cells constantly need to regenerate ATP to sustain life. This regeneration process is called cellular respiration. Cellular respiration is a complex process that involves a series of chemical reactions. The majority of ATP synthesis occurs in the mitochondria. The mitochondria are often referred to as the powerhouses of the cell. The inner mitochondrial membrane plays a crucial role in ATP production. It houses the electron transport chain, or ETC, and ATP synthase. The ETC is a series of protein complexes. These complexes transfer electrons from electron donors to electron acceptors. This electron transfer releases energy. This energy is used to pump protons across the inner mitochondrial membrane. The pumping of protons across the inner mitochondrial membrane creates an electrochemical gradient. This gradient is called the proton motive force, or PMF. The PMF has two components, a chemical gradient and an electrical gradient. The chemical gradient is due to the difference in proton concentration across the membrane. The electrical gradient is due to the difference in charge across the membrane. The PMF is a source of potential energy. This energy is used by ATP synthase to synthesize ATP. ATP synthase is a remarkable enzyme. It acts as a tiny molecular motor. It utilizes the flow of protons down their concentration gradient to drive the synthesis of ATP from ADP and Pi. The discovery of the PMF and its role in ATP synthesis revolutionized our understanding of cellular energetics. Peter Mitchell proposed the chemiosmotic hypothesis in 1961. This hypothesis explained how the energy stored in the PMF could be used to drive ATP synthesis. Mitchell received the Nobel Prize in Chemistry in 1978 for his groundbreaking work. The inner mitochondrial membrane is thus a critical component of cellular respiration. It provides the platform for the ETC and ATP synthase to function. The ETC generates the PMF, which is then used by ATP synthase to synthesize ATP. This intricate process ensures a constant supply of energy for the cell's needs. The story of ATP synthase is a testament to scientific curiosity and ingenuity. In the 1960s, Humberto Fernandez Moran discovered intriguing spherical structures attached to the inner mitochondrial membrane. These structures, later known as F1 particles or F1 spheres, piqued the interest of researchers. What was their function? Ephraim Racker, a prominent biochemist, made a significant breakthrough. He successfully isolated the F1 particles. To his surprise, he found that these particles possessed ATPase activity. In other words, they could hydrolyze ATP into ADP and Pi. This discovery was a crucial piece of the puzzle. However, a critical question remained. How could an enzyme that hydrolyzes ATP be involved in its synthesis? The answer lies in the reversible nature of enzyme activity. Enzymes can catalyze reactions in both directions depending on the cellular conditions. In the case of ATP synthase, the direction of the reaction depends on the direction of proton flow. An ATP synthase is a remarkable molecular machine, often compared to a rotary motor. It consists of two main components, F1 and F0. The F1 component, shaped like a lollipop head, protrudes into the mitochondrial matrix. This component houses the catalytic sites for ATP synthesis. The F0 component, embedded within the inner mitochondrial membrane, acts as a proton channel. As protons flow through the F0 component, they cause it to rotate. This rotation is transmitted to the F1 component via a central stalk. The rotation of the F1 component induces conformational changes in its catalytic sites. These changes facilitate the binding of ADP and Pi, the formation of ATP, and the release of ATP. This rotary mechanism is incredibly efficient, ensuring a constant supply of ATP to fuel cellular processes Understanding the structure and function of ATP synthase has provided invaluable insights into the fundamental processes of life.